Good day, Matrix. All right, so we are going to start with human reproduction specifically. So now we've learned how basic animals go through the process of actually creating more offspring. Now we're going to focus on humans. We're first going to do the structures of humans, or the male and female reproductive organs, and then after that we'll be looking at how the female menstrual cycle works. That is quite a complex section, but it'll be quite a nice section. It's easy. You guys generally find this quite interesting, and it's nice to actually find out how you guys work on the inside, and not just female. We're going to do a little bit of male. We don't do male spermatogenesis, but we do female oogenesis, which is production of an egg, but that's very, very basic. Okay, so we're also going to, as I said, just look at how the cells are produced, specifically in males. So remember that through meiosis, sperm and egg are formed. So we've got a male, he goes through the process of meiosis and produces 4 gametes per parent cell, and then a female, she does the same, but only one of those eggs are actually viable, and we'll talk about that a little bit later. They go through the process of fusion and eventually fertilization. We have a zygote that is produced. Eventually mitosis occurs after the zygote is now diploid from the haploid gametes, and then we have a mass of cells, and eventually differentiation occurs from those different cells, and then we create a new offspring which has a mixture of both male and female DNA. This is the life cycle of humans. I recommend that you just acquaint yourself with it. So please remember, when we did plants last year, we talked about gametogenesis or gametophyte generation, and we spoke about sporophyte generation. What you'll notice over here, that humans have a very short gametophyte generation, and that is only after meiosis, where the sperm cell and the egg cell is produced, and directly after that we go into the zygote, and then we produce a sporophyte generation. That zygote goes through mitosis, we have a growth in number of cells, and then whether you have XX, you become a female, whether you have XY, you become a male. You go through meiosis again, produce sperm, and then the whole thing just goes around. So now we're going to look at the structure of the male reproductive systems. In this, we're going to specifically look at the primary organs, which are the testicles. We're going to look at ducts, so all the tubes, the accessory organs which come off of those tubes and help for the motility of sperm. And finally, we're going to look at the external genitalia, which is known as the penis itself. So let's have a look at the structure of a male reproductive organ. All right, so we're going to start at the scrotum, which is right at the bottom of the middle of the page, and then we're going to work our way through. Please note that I'm going to be following that thick black line, and I'll just be pointing out what is occurring on either side of that. So we have the scrotum itself, which is a sac-like structure that hangs outside of the body, and this scrotum itself holds the testicles. Now remember, we don't have one, we have 2 testicles. And another word for testicle is testis or testes, which is the plural version. On top of the testicle, we have the epididymis, and that is the easiest way to remember this is just the afro on top of the testicle itself. That is where sperm maturation is going to occur. So once it's produced, it's going to wait in over there until ejaculation occurs. From the epididymis, we're going to follow that black line, and please notice that it goes all the way in front of the pubic bone. Okay, so it goes up directly. This is in the system itself. In front of the pubic bone, it's going to go around the bladder, and once this goes around the bladder, it is then going to meet the seminal vesicle. Now, that black line that you see is called the vas deferens, okay? And you'll see that label at the top left-hand corner. So the sperm is going to be created in the testicle, it's going to mature in the epididymis, it's then going to move all the way up the front part of the body, all the way around the bladder, up the vas deferens, and eventually it's going to be met with the seminal vesicle. The seminal vesicle is very close to that large apple-like structure over there called the prostrate gland. This is what stimulates a whole bunch of stuff in the body. It doesn't produce testosterone, that happens in the testicles itself, but it helps add a whole bunch of liquids that are very useful a little bit later. Now I want you to notice something extremely weird, because the tube that is coming out of the bladder and the vas deferens that is meeting in the prostrate gland all meet into one, which means that men do not have 2 separate tubes, one for urine and one for sperm. Also, we'll differentiate the difference between semen and sperm a little bit later. Please also note that each testicle has an epididymis and a vas deferens, and these two tubes need to meet. And that meets just by the seminal vesicle over there, and that is called the ejaculatory duct. Once it moves through the prostrate gland, it's going to be met by the Cowper gland. The Cowper gland sits at the base of the shaft of the penis, and that is also going to secrete a whole bunch of very important substances. Eventually, this vas deferens now becomes known as the urethra as it goes through the shaft of the penis, and eventually gets ejaculated out of the glands or the head of the penis. Either word is acceptable. Also, the foreskin surrounds the head as well, and there are two different words for this. You can either use the word prepuce or you can use the word foreskin. In an exam, I think it was last year's or the year prior to that, they actually asked for where the position of the foreskin is. So please note, you can either write at the base of the head or over the head. We'll go over the definition a little bit later. What I then want to bring up to you is along the shaft of this particular penis, you can notice there are two other words, the corpus spongiosum and the corpus cavernosum. There are two spongy muscle tissues that run along the top part of the penis, and that is where blood enters and causes the erection of the penis, as well as the corpus spongiosum, which also, please notice that it's in that entire chunk all the way to the head of the penis, and the urethra runs through the corpus spongiosum. All right, please notice this is also directly next to the anus, so it's not like these are very far apart, and that is why when a man has potential prostate cancer or prostate problems, you can go through the anus and you can actually feel the prostate over there and figure out what is going on. Please also note that the pubic bone occurs both in male and female, okay? It's part of your pelvis. This is the front view of the male reproductive organ. So let's go the opposite way. So we'll start at the gray part in the middle called the glans penis. Around that is the foreskin. So remember, the glans is also known as the head. Then we have the foreskin, also known as the prepuce. Then we have the penis itself, which has spongy tissue, specifically spongy muscle that can be pushed full of blood, and that causes the penis to become erect. Then as we go further up, we find the Cowper's gland. Then we split up, so now you can see that there are two separate tubes that go each one to a testicle. So after the Cowper gland, we enter the prostrate, which is that apple-like structure over there. We go past the seminal vesicles, up over the ureter, okay, that is coming from the kidneys, and then that is going all the way down. Once it goes over that, it's now known as the as the vas deferens. The vas deferens then meets the epididymis, which looks like an afro on top of the testicles on either side. And please remember, around each and every single testicle, we find the scrotum. We'll talk about it a little bit later, but note that like a male's testicles actually move up and down either into the body or out of the body, and that is because sperm can only develop at very specific temperatures, and we'll talk about those a little bit later. I recommend you pause the video over here and you draw this, please. This is the front view of a penis. If you would like to draw the previous one, you can also draw that. Either one is perfectly fine. This one is maybe just a little bit more simple, and it also looks like there's a clown's afro around the testicles. Please note that the skin, however, is folded quite a bit, and this allows for the movement of the testicles up and down so that the skin doesn't tear if it needs to drop too far. So let's talk about the testicles themselves. They are the male primary sex organs, and I've included a longitudinal section of the testicles. So let's have a look at the longitudinal section before we continue with what it does. So inside of the testis itself, we have the seminiferous tubules. Now these tubules can go anywhere up to about 250 meters per testicle. All right, so the tubes are extremely convoluted, and they really do allow for a large amount of production of sperm. So if you increase that surface area, it means that you're going to increase the amount of cells that are able to produce sperm. From the seminiferous tubules, you can see that it enters the epididymis, and here's where the sperm sits and matures and grows until it is ready. And then once it is ready to be moved, it will then be activated by the accessory organs a little bit later. So there you can see that vas deferens, and then we call it the spermatic cord. I'm not worried about that. So what is the testes responsible for? They're responsible for producing spermatozoa or sperm. So remember, the word zoa means of animals, so sperm of animals, in this particular instance sperm. So the process of producing sperm is called spermatogenesis. So genesis means the beginning of, or the creation of, and sperm, so the creation of sperm. It occurs in the scrotum outside of the abdominal cavity, and the sperm require temperature lower than the body temperature. So you need to know off by heart that sperm production, the optimal temperature is between 35 to 36 degrees Celsius. And this is why men's underwear has to be extremely breathy, because the testicles need to be able to move. Otherwise, they will suffer from infertility if the temperatures are not correct, if they're either too hot or too cold. If they're too hot, the scrotum itself will drop, causing the testicles to be further away from the body so that there is some sort of air that moves over it. And if it is too cold, the testicles will move inside of the cavity, inside of the male body, and that will be able to keep them warm. Within each testicle, the seminiferous tubules are found, and those seminiferous tubules are lined with two specific kinds of cells. The first kind is the Sertoli cells. If you want to remind yourself how to, like a quick reminder, just remember Sertoli or Sertoli, depending on how you used to pronounce it. So the Sertoli cells or Sertoli cells are found in the seminiferous tubules. They actually line the inside of the tubules themselves. They are extremely rich in glycogen. Now remember, when we did the pancreas, you remember that glycogen is actually the stored form of glucose. So it is a very energy rich sort of fat, and this fat is going to be used to propel the sperm cell well into the female's reproductive organ. So glycogen provides nutrition for the spermatids. So spermatids are baby sperms as they develop into sperm cells, and we'll look at what diagrams of that later. At puberty, the germinal epithelium start to produce sperm cells by spermatogenesis. So this is the second type of cell that you also find lining the inside of the seminiferous tubule. Seminiferous tubule, and these actual germinal epithelium is what goes through the process of spermatogenesis to produce the cells. Now, in between the seminiferous tubules are the cells of Leydig, okay, or Leydig, depending how you feel like pronouncing it. This is extremely important. So the cells of Sertoli, germinal epithelium, and now between the seminiferous tubules are the cells of Leydig. They secrete the sex hormone testosterone, and please remember this is responsible for the growth of hair or facial hair, hair in the armpits, pubic hair, lowering of the voice, development of muscles, okay? And this starts happening between the age of 11 to 15. What we're starting to find nowadays is that boys are maybe dropping to about the age of 9/10 years old, and then you also get some anomalies where some boys only start hitting puberty around the age of 16, 17 years old. But this really just depends on the child's development as a whole. So I've included a diagram over here. You can also find these diagrams in your textbooks, but I just prefer this as well. If you'd like to draw this, maybe just draw a small little portion of it, and then we'll be able to then you'll just insinuate that the rest of the cell looks the same. So when you draw this, please label the Sertoli cells, spermatids. Don't worry about the secondary spermatocyte. Don't worry about the primary spermatocyte, and don't worry about the spermatogonium. Okay, I'm not really too phased about them, but if you remember, well, like if you would like to label the spermatogonium, rather label it as the germinal epithelium. Okay, so what do the Sertoli cells do? They're within the seminiferous tubules, and they're rich in glycogen, and the glycogen provides nutrition for the sperm. Finally, we have the Leydig cells, and they are found between the seminiferous tubules, and they secrete testosterone, and they are directly responsible for puberty. So I was a bit confused about the Sertoli cells, so I went and found another diagram so that you guys can maybe see a bit better. So we have the spermatogonium at the top left-hand corner over there. Please note that this is also known as the germinal epithelium. And directly next to it, we have the Sertoli cells, or Sertoli cells, or Sertoli cells, depending how you feel like pronouncing it. Note that once the spermatogonium have actually gone through the process of meiosis and started producing the spermatids, they attach to the, well, they don't really attach to the Sertoli cells, but they kind of move onto the Sertoli cells so they can actually get a whole bunch of glycogen. Let's have a look at the tubes responsible for carrying the spermatozoa. So I've included a diagram over there, and I've put red blocks around the 4 specific important bits. So we have the epididymis. This is from the seminiferous tubules to the vas deferens, and it temporarily stores sperm. Please add in there that it's also used for the maturation of sperm, and that is that afro-like structure on top of the testes. We then have the vas deferens, which carries the sperm from the epididymis through the abdomen into the ejaculatory duct. Okay, and remember the ejaculatory duct is what joins the urethra after it leaves the bladder, and the contraction of the walls of the ejaculatory duct force semen through the urethra. So they are extremely strong, not extremely strong, but there are muscles that go around the ejaculatory duct which are able to contract extremely and actually force all of the semen directly out. So the difference between semen and sperm is only the cell itself. Semen is a mixture of all the different secretions from the accessory glands, which we will go through, including and plus the sperm. Okay, so semen is sperm plus secretions, whereas sperm is just the cell itself. Okay, so please note that the ejaculatory ducts are there for the contraction and allows for ejaculation to occur. Finally, we have the urethra. This is a tube running through the penis. Remember, specifically the one that runs on top of the at the base of the penis, and this is a common tube for urine and semen to pass through. They like asking that for some reason. You know, what is the difference between male and females? Females have 2 separate holes, one for urine, one for sexual reproduction, whereas males have one tube for both. Now we're going to look at the accessory glands. So you'll find your seminal vesicles. They secrete extra fluid as sperm passes through the vas deferens. Okay, this contains nutrients, enzymes, and hormones. So this is where we're going to start creating movement within the sperm, because until now the sperm in the epididymis that's been moving up the vas deferens are not moving themselves. The body itself is pushing that sperm up the vas deferens. Once it gets past the seminal vesicles, that is when we are then going to start moving or allowing the sperm to become more active, as it gives the sperm energy. The fluid neutralizes acidity from the urine because the sperm needs alkalinity. So if the urethra or the vas deferens is too acidic, the sperm will die before ejaculation can occur. So we need to neutralize that acidity and actually add more substances to create a more alkaline environment. Then we have the prostrate gland, which secretes 1/3 of the fluid, making up the semen. So this contains enzymes which make the sperm more motile. So now we're kicking into mode now. So now you want to make sure that the sperm are actually moving, because if those hormones are not secreted before they get past the prostrate gland, if ejaculation happens too quickly, the sperm itself will not be able to swim up the vaginal canal and eventually into the fallopian tubes. Then we finally have the Cowper gland, which is very much at the base of the penis. This secretes mucus, which has a lubricating effect, and this also decreases the acidity of the urethra. Okay, so the lubricating effect is because there's potentially is too much friction between the sperm and the urethra, which might stop all the sperm from actually being ejaculated. So creating a lubricating effect is the equivalent of throwing soap on a slide, so that you are able to move through a lot quicker. I recommend you draw this. This is a very simplified view of from the testicles all the way through the penis. So we have organs on the left and functions on the right. So these is a quick little summary of everything we've done. So the testes with the seminiferous tubules allow for sperm production. We then have the collecting duct, which are moving into the epididymis for transport and storage. Finally, the epididymis, which is for transport, maturation of the sperm, and ejaculation. So it was just the movement of this is when ejaculation actually starts to take place, as soon as it moves out of the testicles itself. Then we have the vas deferens, also known as the sperm duct. And please note that the vas deferens is the tube that we cut and or tie when a man goes for a vasectomy. Because if we cut that tube, that ultimately means that sperm cannot get from the testicles through out of the urethra for ejaculation. So if we do that, we can stop, or we can kind of make sure that the man is not going to be ejaculating any sperm. He will still produce all the secretions from the accessory glands, but it will just not contain any moving sperm or any sperm whatsoever. Then we have the seminal vesicle. This secretes a thick liquid to transport the sperm. We have the prostrate gland, which secretes this thin alkaline solution to neutralize the urine and female system, because remember the vaginal canal should also have a very specific pH, and if that pH is incorrect, even if the pH is correct within the male's tubules, the sperm will not survive as it enters the female reproductive organ or the vaginal canal. Finally, we have the Cowper's gland. Secretions may lubricate or flush out urine to form a gelatinous plug. Yes, sounds gross. The urethra, passage for the urine and sperm, and finally we have the penis, which is used for copulation. You can use whatever word you'd like, so copulation, coitus. Don't just use the word sex, because remember the word sex is like how you sort of recognize as a male or female. This is a fun little picture of Kanye West and a cross-section of a penis. You can notice that there's two spongy tissues right at the top, and then we have one directly running through the bottom where the urethra is going through. So this is a cross-section through it. I've never seen this in a test or an exam, but maybe draw it for yourself. I really haven't seen many tests have it inside, but maybe it's just more for you guys to learn. So we have the dorsal veins and nerves. Remember that there are tons of nerves that are running through this, and it's important for nerves to also run through, because the body needs to be able to recognize when it is actually going through the process of copulation. We then have a collagenous sheath. This is to create a lot of structure. Finally, we have the corpus cavernosum. There are two on either side of the penis, and they have a central artery. And this is going to secrete a lot, well, not secrete, yeah, it is secrete technically, a lot of tissue fluid. So it's not like your blood is escaping. It just means that the tissue fluid is going to move into the corpus cavernosum and actually cause those spongy muscles to become turgid, remember from plants, or the opposite of flaccid, so they become extremely rigid, like hard. And then finally we have the urethra, which is running through the corpus spongiosum, and that is going to create a very specific and what is the word I'm looking for? Open line for ejaculation to occur. So it says a spongy tissue fills with blood when aroused and transfers sperm from male to female. Finally, right at the top over there, the foreskin covers the tip of the penis. You are also allowed to use the tip of the penis in a test or an exam. You don't have to use the word glans or head. Can be removed for religious or cultural or health reasons. And there are a lot of instances where sometimes the foreskin, when you are developing, just doesn't grow big enough around the glans, and that actually needs to be cut off. And this is called circumcision. Please don't confuse female circumcision with male circumcision. They are two completely different things, and maybe we'll talk about that when we get to school. It's a bit of a touchy subject. All right, now we need to look at, once the sperm eventually gets out, how does it look like? Okay, so now it's moving. We have the tail for swimming. It has mitochondria to supply energy for swimming, and this is called the middle piece. So what attaches the head to the tail is the middle piece. Please also note that there are no mitochondria in the head of the sperm itself, which means that that's why you only get mitochondria from your mother, and that's why we call it mitochondrial DNA. And the mitochondrial DNA only comes from the mother, because there's no mitochondria in the head itself, so that it can't get in the way when fusion is eventually going to occur. So in the head itself, we have the nucleus, which is haploid. Remember, that means that you only have one set, not two sets of DNA. And then we finally have the acrosome, which is right at the head. The acrosome is the most important part. So it produces enzymes to dissolve the surface of the egg. Remember that there's going to be a gelatinous substance that is surrounding the egg, because you don't want it to dry out for unnecessary reasons. So the acrosome is going to open up and produce these digestive enzymes, which are going to eat its way through the gelatinous substance and allow for the fusion to occur. And only the head is going to enter into the female, the egg itself, which is going to then allow the nucleus to move out and allow for fertilization to occur. So now here's an annotated diagram of structure of a sperm. I have seen this in a test where they've asked you to draw it. Most times they just ask you to label it. This is on page 97 of your textbooks, okay? And again, we have the head, The head is made out of the nucleus and the acrosome, which contains hydrolytic enzymes, which just digests a layer around the actual egg. Then we have the middle body, or the middle piece, which is known as has the mitochondria inside, which provides energy for movement for the tail. And notice there are a lot of mitochondria. So all their glycogen is going to be broken back down into glucose. That is going to move into the mitochondria, go through a lot of cellular respiration, and release tons of adenosine triphosphate, which eventually is going to attach to the tail. And the tail is made out of contractile fibers. Now, these contractile fibers are actually proteinaceous, or they're made out of protein fibers, mainly actin and myosin, and they're actually going to contract and release in such a specific way that it's going to cause the muscle fibers to move. And this is responsible for a whip-like movement of the tail. All right, so I hope you guys coped through all of that and saying a whole bunch of the word penis. If you have any questions, maybe ones that I don't really want to talk about in this lesson, you are more than welcome to WhatsApp me. All right, have a good day, ladies.